Hello everyone, welcome back to my channel Pari's tutorial. I'm back with one more interesting video. So let's get into the class. Last class we have discussed about the types of RNA. What are the types of RNA? rRNA, mRNA and tRNA, their structure and their function. Okay. Now this class I want to discuss about the process called protein synthesis. So what is protein synthesis? Before you think about protein synthesis, we have to have a clear idea about transcription and translation. Guys, you have already gone through the DNA replication, how the DNA transfers the information material. Okay, what is the mode of replication and all in the previous class itself. Today, this time, I want to concentrate on transcription. What is transcription and how you'll get an mRNA from DNA. Okay, so let's start the class. So this in this class we have to know that what is central dogma. Central dogma is nothing but the information of the DNA has to be carried to the RNA, and from RNA it gets translated into proteins where we get a polypeptide chain. Okay, that information is being transferred from DNA to RNA and RNA to proteins. So this is the central dogma, and it was first proposed by Crick. The information present in the DNA is transferred to DNA by replication and to RNA by transcription, and from RNA by protein translation. That is what you call the central dogma. Okay, so the process of production of RNA copy of a DNA is called transcription. Okay, already I've gone through the replication. Now what is transcription? You will get an mRNA called messenger RNA from a DNA. What does that mean? In transcription, the DNA has got that DNA has got two chains in it, right? So out of which one is called a sense strand, the other is called the antisense strand. Okay, so the antisense strand would act as a template so that you will get a sense mRNA. Okay, sense mRNA is produced from the antisense strand of the DNA. Based on it, the complementary bases have been based on it. So obviously in RNA you will be getting uracil instead of thymine, that you already knew. Okay, the strand that is produced from mRNA is complementary to the antisense strand of the DNA. That is what you call this transcription. Let's see what is really transcription in, is what are the process involved in it. Okay, and this reaction is catalyzed by RNA polymerase. This I have. What is a transcription unit? Okay, the transcription unit has got a promoter sequence. See, let's see, this is your start point of your transcription where the RNA polymerase has to bind. For example, this is the DNA, double stranded DNA. Okay, I'll draw it better. Okay, that would help you for a better understanding. Okay, 5 prime side and 3 prime side of your DNA, and then this is your right. So this particular strand acts as an antisense strand. Okay, and due to which the RNA polymerase would bind and produce a mRNA, which is a sense RNA. It has got complementary bases with the antisense strand of this DNA. Right? This is primarily, you call it as a transcription. For that to happen, the RNA polymerase has to bind to the DNA complex. So, for that, you have to understand what is a transcription unit. So, transcription unit is a stretch of sequence of DNA that is transcribed into single RNA molecule. A typical transcription unit has a promoter. Yes. See, let me draw about a prokaryotic promoter so that you will get an idea about it. I'm so sorry for the inconvenience. Restore soon. Okay, so let's start the prokaryotic promoter. This is your DNA. Okay, and this is, you feel, this is your start point. Generally, the start point is purine rich. Okay, so here this is where the transcription starts and moves in this direction. Okay, so from here you will call it as upstream sequences and downstream sequences. Okay, so based on it, say this is the whole promoter. Promoter is nothing but the point here to before the initiation starts is called promoter. These promoter sequences have got two different sequences in prokaryotes. Okay, see, first one is being -10 sequences upstream, the other one being -35 sequences. Okay, the promoters generally have, this is a common sequence for this. So obviously this is a sequence for the sense strand of DNA, and the antisense strand would be, right, this is your -35 sequence. And here in the 10th here the 10 base pairs from your start point, you got a sequence called, this is AT rich. Why is this AT rich? Because for the DNA to form an mRNA, for the production of mRNA, the two DNA molecules should get melted so that the antisense strand slightly separates from the sense strand, and this antisense strand would act as a template. For that to happen, the promoter sequence should get a sequence of AT, so that here the RNA polymerase would bind, and easily the AT rich needs lesser energy, lesser GTP or ATP energy for melting of your double stranded DNA, so that it would be easy for you to transcribe into mRNA. Okay, so this is called the AT rich site, and this is -35 sequence site. Okay, so in the in middle you have got a base pairs of 16 base pairs, like it's like dispense like in this. Okay, what is the function of this particular sequence? This particular sequence is meant for the recognition. So this promoter region, it will attract the RNA polymerase. RNA polymerase would bind to this sequence. This 35 sequence code would recognize, this particular 35 sequences would recognize the RNA polymerase and helps for the binding of it. Okay, and this keeps on moving in this direction for the transcription, and hence you will get mRNA. So this is the promoter unit for prokaryotes. Okay, so let's go to, this is what I am talking about, a start point, a coding region, and termination sequence. So this is the transcription, and I already taught you in the with the diagram. Okay, so next, so after transcript is bacterial RNA polymerase. It is an RNA polymerase which binds to your double stranded DNA for you to transcribe, right? In most of the bacteria, a single type of RNA polymerase is found. Okay, and the complete RNA polymerase molecule is called holoenzyme. Okay, see, the RNA molecule is a big RNA molecule. RNA polymerase is a big thing in bacteria. It has got a core enzyme and a sigma factor in it. So here comes the important topic. So once you got your DNA, so what happens is the transcription happens in nucleus. Okay, once the transcription happens, you will get mRNA. That is what the primary transcripts. These primary transcripts are very unstable, so they either should get degraded, or they should undergo splicing, or they should undergo further changes. So for the production of mRNA, eukaryotic mRNA, for eukaryotic rRNA and eukaryotic tRNA to happen, they should undergo some changes. So they itself can't participate for the translation. They have to undergo splicing, or they have to have a poly A tail addition and 5' cap site addition. These are all the things that happen. There is what we call RNA editing. Okay, mRNA of eukaryotes would never immediately enter into cytoplasm for the translation to happen. They have to undergo many changes like addition of 5' cap, addition of poly A tail, removal of all the introns, and joining all the exons, and these are the things that generally would happen for eukaryotic mRNA. Okay, so I have told you the translation begins always from 5' to 3'. In terms of polypeptide being synthesized, it starts with N-terminal polypeptide, while C-terminal. You know that N is a amino terminal part of your amino acid chain, and C terminal is the carboxy terminal of your amino acid. So it when the translation moves in 5' to 3' direction, the M terminal, the protein synthesis or the protein chain would start with N terminal and finally ends with the C terminal. That is how the whole part is like. Okay, so what is the process involved in translation? It obviously have three different process: initiation, elongation and termination. Okay, so in prokaryotes, what happens is there is no RNA editing happening. So whatever the RNA that is produced immediately goes for the translation. Okay, but for eukaryotes they have to undergo many changes. Okay, so what happens in initiation prokaryotes? So I have given you a small diagram that would be easy for you. See, these are the initiation factors. So you all know that in prokaryotes the ribosome complex is a 70s subunit, right? It has got two important 30s and 50s subunits, a smaller subunit and a larger subunit. The IF3 is the initiation factor 3 would bind to it and would stabilize your ribosomal 30s complex. Okay, and after that it will get attached to the mRNA region. Okay, and to get ready for your translation. So for that, in meanwhile, formal methionine tRNA is the RNA. Sorry. Formal methionine tRNA is the tRNA that codes for formal methionine in prokaryotes. So this one, along with your IF2, which is nothing but initiation factor 2, would form a binary complex. That binary complex would, along with the GTP, would form a initiation complex where formal methionine would bind to the codon region. Codon region is AUG, right? AUG is the initiation codon for translation to happen. So you have already gone through that the ribosomes got two important sites, P site and A site. The first initiation codon AUG would directly go to the P site of your ribosomes. The additional tRNAs would keep adding to your A site, but the first initiation codon that have the anticodon for your mRNA would be directly binding to the AUG codon of mRNA. So that codes for formal methionine in prokaryotes. Okay, so once this initiation complex has happened, everything gets stabilized, GTP gets cleaved, and IF3 and IF2, both initiation factors, would be removed, and finally you are done with your translation. Okay, and the termination would happen only when the mRNA, only when the tRNA identifies the nonsense codons. So once they reach to the nonsense codons, there are no amino acids specifying that particular codon on mRNA, and thus the two ribosomal subunits gets dissociated and the polypeptide chain that is growing on get removed, and the tRNA would get removed from your P site. So that I will tell you in elongation process. So this is what, see, this is the eukaryotic factor. You already know that eukaryotic mRNA has got a 5' cap and a 3' poly A tail after modifications. Okay, so this particular 5' cap mRNA has got an elongation factor. These are all the elongation factors. Okay, this elongation factor 4E would bind to the mRNA, making it stabilized. Finally, it has got A, B and E. These are the three different elongation factors that stabilizes your mRNA for your translation. In meanwhile, the methionine tRNA, along with the elongation factor 2 and GTP, makes a binary complex again and forms a ternary complex, finally with a 40S subunit. So in eukaryotes, it's a 80S ribosome. So it has got two subunits, one larger 60S and smaller 40S. So the every ternary complex binds with the 40S subunit. Okay, so once it binds with the 40S subunit, it is ready for the initiation of translation. So for that to happen, the 60S ribosome would get binded to it and the process would happen. So the methionine tRNA would directly enter to the P site of your ribosome. Okay, where it codes for AUG again in eukaryotes. Okay, so this keeps on moving, and finally at the end you will get a chain of polypeptide molecules on the P site itself, because in the elongation I will let you know how the chain is formed. Okay, so this is the process of protein synthesis. See, this is a 30S ribosomal particle P and A. I told you this is your P site and this is your A site. So formation of the initiation complex. Okay, the tRNA which has got formyl methionine. This is your in prokaryotes. Okay, that is why they have mentioned it particularly. It's formyl methionine tRNA, which is having an anticodon to the codon of your mRNA, right? So it directly gets attached to the P site of your ribosome. Then attachment of your 50th ribosome would happen, and once it happens, it forms a peptidal site, right? So now once this is moved, this based on the sequence present on the codon region of your mRNA, the entry of your amino cell tRNA would happen. Okay, see, the ribosome moves once in three times, like once in three codons it will move, like once in three bases it will move from left to right. So based on this, when it moves from AUG to CUG, another vacancy would happen in the A site, which attracts the amino acid tRNA that is anticodon of your mRNA, right? So if it is CUG here, it would be GAC here. So finally the leucine amino acid would attach here, and hence the polypeptide chain is again growing. So binding of tRNA and elongation factors would happen, and after that there is a formation of peptide bond. Once there is a formation of peptide bond, this formyl methionine gets moved to the over the leucine, and hence the whole amino cell tRNA with two amino acids would keep moving to P site. So making making A site available for another amino acid. So this is how it happens. First it happens in the P site, then A site is attracted, and then whatever the amino acids that are formed on the P site would be attached to the A site, and from A site the whole tRNA moves towards the P site, making A site available for the other amino acid tRNA, right, based on the codon of mRNA. So this keeps on moving and the chain keeps growing. Okay, the chain keeps growing in the A site, keeps elongating in the P site of your ribosome. So this is how the polypeptide chain is formed. Once the ribosome enters into the final codon, see, this is how the termination happens. Continuation of previous steps for each mRNA, and now it enters UAA. I told you UAA is an anticodon. Sorry, UAA is a terminating codon. It doesn't specify for any amino acid. So binding of termination protein release factors, so due to which you got a dissociation and complete linkage of protein polypeptide chain. So hope you got an idea about how the movement of translation happens, how mRNA gets transcribed, and finally how the proteins have been translated in a gene. So this is all about the protein synthesis that I want to talk about. UAA, UAG and UGA, and is also mediated by certain release factors. So when the A site of ribosome reaches a nonsense codon, the polypeptide chain detaches from the tRNA located at the P site. Immediate release of tRNA from the P site and the release of ribosome from the mRNA. So whatever is attached, the whole thing goes away. So this is how the translation happens. Okay, this is, hope you got an idea about protein synthesis. Hope you are clear with this. Okay, thank you and have a nice day. See you in the next video. Thank you, and please subscribe my channel and please like, comment and share it. Thank you.